welcome back. Tonight, we're going somewhere genuinely cold. Specifically, to the continental shelf waters around South Georgia Island, at the edge of the Southern Ocean, at 2 in the morning in the middle of Antarctic winter. The water temperature here is minus 1.9 degrees Celsius, just above the freezing point of salt water. The surface is sealed under pack ice. The benthic plain below is in complete darkness. And resting on the seafloor, entirely motionless, is a fish with no red blood cells, a transparent body, and a heart the size of a mammal's. Take a slow breath. Let your body settle. We're going under. The continental shelf of South Georgia is not a featureless void. It is a textured landscape of soft glacial muds, scattered basalt boulders dropped by carving icebergs, and dense communities of cold-adapted invertebrates. Glass sponges, their skeletons made of pure silica, draw water through their chambers with an almost imperceptible flow. Long-legged sea spiders move so slowly across the substrate that they appear stationary. Pale, brittle stars blanket the mud in overlapping geometric arrays. The water above the benthos is hyper-oxygenated. At minus 1.9 degrees, seawater is extraordinarily dense. The molecules pack tightly together, increasing the solubility of dissolved gases well beyond what warm water oceans can hold. When winter winds drive cold surface water downward in the ventilation process that oceanographers call deep water formation, they flood the benthic plain with a continuous descending current of oxygen-rich fluid. The seafloor here is not stagnant. It is continuously refreshed from above. In this environment, resting with broad pectoral fins spread lightly across the mud, is Chaenocephalus aceratus, the South Georgia icefish, a member of the family Chaenocephalus. It is translucent. Through its colorless chest wall, visible in the beam of a submersible light, the slow contraction of its heart is directly observable, a large pale pump beating with a cadence that matches the deep, unhurried rhythms of the polar night. Every other vertebrate on Earth, every fish, every amphibian, every reptile, bird and mammal uses the same basic strategy for oxygen transport. Red blood cells packed with haemoglobin, an iron-bearing protein that binds oxygen in the gills or lungs and releases it into working tissue. The system is ancient, conserved, and essentially universal across the vertebrate lineage. The icefish has none of it. Draw a blood sample from an icefish, and it looks like water. Thin, colorless, completely clear plasma with no cellular cargo whatsoever. It is the only known vertebrate on Earth that entirely lacks red blood cells as an adult and produces no hemoglobin at all. Its blood carries no iron, no pigment, no molecular oxygen carrier. Instead, it relies on a physical property of its environment. Cold liquids dissolve gases more efficiently than warm ones, the same reason a cold carbonated drink holds its effervescence longer than a warm one. The near-freezing waters of the Southern Ocean hold nearly twice the dissolved oxygen of tropical seawater. Because the icefish lives in permanent contact with this oxygen-saturated fluid, and because its metabolic demands are extremely low in the cold, oxygen dissolves directly across its large gill membranes into the blood plasma as free-floating gas, and travels to tissue through simple fluid dynamics rather than active molecular transport. The oxygen doesn't ride a carrier, it drifts. The loss of hemoglobin was not a gradual, carefully managed evolutionary transition. It was a deletion, a massive genomic accident that should, under any other conditions, have been immediately fatal. Approximately 5 to 6 million years ago, the Antarctic circumpolar current was fully established, having formed when the Drake Passage opened between Antarctica and South America around 30 million years ago. This current created a permanent barrier between the Southern Ocean and the warmer seas to the north, trapping the ancestral fish of the region in water that was cooling rapidly toward freezing. Under this pressure, in the common ancestor of what would become the 16 known icefish species, large blocks of DNA encoding the hemoglobin protein subunits were deleted from the genome. In 15 of the 16 species, the gene for the beta subunit is entirely absent. A single non-functional fragment of the alpha subunit persists as a pseudogene, a molecular remnant with no protein product, a silent relic of what the organism used to be. In any warmer ocean, this mutation would have caused immediate suffocation. A red-blooded fish in the Atlantic or Pacific would die within minutes if its hemoglobin production ceased. But the ancestral icefish was not in a warm ocean. It was in the one environment on Earth where the ambient dissolved oxygen concentration was high enough and the metabolic rate cold enough that the loss of the oxygen carrier was survivable. The deletion occurred in the right place, at the right time, in the right direction. A catastrophic mutation became a founding event. The fish drifted into a new physiological state, not because it was selected toward it, but because the environment was cold enough to tolerate the accident. Carrying oxygen in simple solution, rather than bound to hemoglobin, reduces the oxygen-carrying capacity of the blood to roughly 10% of what red-blooded fish achieve per unit volume. To compensate, the icefish completely redesigned its circulatory infrastructure. The first compensation is volume. An icefish carries between 2 and 4 times the blood volume of a comparably sized red-blooded fish. Even though each milliliter carries less oxygen, the sheer quantity of fluid in constant circulation maintains a sufficient supply to the tissues. The second compensation is the heart. The cardiac mass of an icefish is up to 3 times greater than that of its red-blooded relatives, comparable in relative size to a mammalian heart. This enlarged pump doesn't beat rapidly. It beats slowly and powerfully, delivering enormous stroke volumes with each contraction. The strategy is not speed, but displacement, moving large sheets of dilute plasma through the body rather than small volumes of oxygen-dense blood. The third compensation is vascular diameter. Because clear plasma has no red blood cells to create viscosity, it flows far more easily than conventional blood, especially in the cold, where red blood cells in other species would stiffen and aggregate. The icefish exploits this by widening its blood vessels significantly. Larger diameter vessels reduce hydraulic resistance according to the fourth power relationship of Poiseuille's law. Doubling the diameter drops resistance by a factor of 16. The icefish pumps a vast, thin, low-viscosity fluid through wide channels at low pressure, achieving whole-body circulation at a fraction of the energy cost of a conventional circulatory system. The result is among the lowest resting blood pressures ever recorded in a vertebrate. In several icefish species, most notably Chaenocephalus wilsoni, the blackfin icefish, the process of genomic simplification extended further than hemoglobin. A separate, independent mutation silenced the gene for myoglobin, the oxygen-storing protein of muscle tissue. In most vertebrates, myoglobin gives the heart its characteristic dark red color. It buffers oxygen within cardiac muscle, releasing it during periods of high demand when the blood supply can't keep pace. In the blackfin icefish, the heart is not red. It is not even pink. It is white, a pale, translucent pump made of muscle fiber containing no myoglobin at all. The white cardiac muscle relies entirely on oxygen diffusing directly from the clear plasma passing through its chambers. For this to work at all, the plasma must remain highly saturated with dissolved oxygen at all times, which it does, because the Southern Ocean water flooding the gills is already close to equilibrium saturation, and the fish's metabolic demands remain low enough that the tissue never significantly depletes the supply between heartbeats. Two independent genetic events, millions of years apart, both eliminating iron-based oxygen proteins. The icefish is not simply missing hemoglobin. It has systematically shed the entire iron-dependent architecture of conventional vertebrate oxygen management. The water temperature at -1.9 degrees presents a direct physical threat. Standard vertebrate body fluids freeze at approximately -0.8 degrees Celsius, well above the ambient temperature of the Southern Ocean. Without intervention, the icefish's blood would crystallize, forming ice needles that would rupture cell membranes throughout its body. The solution evolved long before the hemoglobin deletion. It was, in fact, the prerequisite that made survival in these waters possible at all. Approximately 30 to 40 million years ago, as Antarctic waters began their long cooling trajectory after the opening of the Drake Passage, a gene encoding an ordinary pancreatic digestive enzyme underwent a series of duplication and mutation events under intense selective pressure. The duplicated copies accumulated mutations that altered the protein's structure, eventually producing an entirely new molecule, the antifreeze glycoprotein. The mechanism by which these proteins work is not chemical depression of the freezing point, the way salt works. That would require concentrations high enough to be osmotically lethal. Instead, the antifreeze glycoproteins function through a process called adsorption inhibition. As microscopic ice crystals inevitably enter the fish's body through the gills or across the skin, the antifreeze proteins, circulating continuously in the clear plasma, bind directly to the faces of the crystal lattice. By coating the growth surfaces of the developing crystal, they prevent water molecules from attaching to extend it. The ice cannot grow. It is arrested at a size too small to cause tissue damage. The icefish carries these immobilized microcrystals within its plasma indefinitely, eventually routing them to the spleen for disposal during periods when the fish moves to slightly warmer water. This means the icefish is never entirely ice-free internally. It lives in a permanent state of managed ice, carrying frozen matter in its blood, kept under molecular arrest by a protein that the genome repurposed from a digestive enzyme more than 30 million years ago. The final compensation in the icefish's oxygen economy is subcellular. In the muscle cells of a typical temperate fish, mitochondria, the organelles that convert oxygen and glucose into ATP, occupy roughly 5 to 10% of the cell volume. In the icefish, this figure reaches 30 to 35%. The cells are packed with mitochondria at a concentration found in no other fish species examined. Crucially, these mitochondria are not distributed randomly through the cell interior. They cluster immediately adjacent to the cell membrane, directly beneath the capillary walls. This positioning minimizes the diffusion distance between the plasma carrying dissolved oxygen and the organelles consuming it. Because there is no hemoglobin releasing oxygen at specific delivery sites, the gas must travel by passive diffusion from the blood across the cell boundary. Every micrometer of additional distance reduces delivery efficiency. By moving its mitochondria to the perimeter, the icefish eliminates most of that distance. The combined effect is a metabolic architecture calibrated entirely for low-rate, sustained, efficient operation. The icefish cannot generate rapid bursts of speed. There is no system capable of supplying oxygen fast enough for that. What it can do is maintain a continuous, reliable baseline of energy production across the long, dark months of Antarctic winter, in complete darkness, at temperatures where most biochemical systems would slow toward failure. It doesn't need speed. Nothing it competes with here has any either. The genomic consequences of losing hemoglobin extend into metabolism in ways that are not immediately obvious. In all red-blooded vertebrates, iron is a carefully managed and tightly recycled resource. It is chemically reactive when unbound. Free iron generates destructive hydroxyl radicals through the Fenton reaction, damaging lipids, proteins, and DNA. To prevent this, vertebrates maintain a sophisticated network of transport and storage proteins. Transferrin carries iron through the blood, ferritin stores it safely in tissues, and the liver and spleen continuously recycle iron from aging red blood cells back into new hemoglobin synthesis. The icefish has no hemoglobin to recycle and no significant iron demand. Over millions of years of isolation, the genes governing iron absorption, transport, and storage have drifted progressively toward dormancy. The icefish does not seek iron in its diet. It does not maintain the metabolic infrastructure required to handle it. The result is a cellular environment essentially free of the iron management burden that represents a significant ongoing metabolic cost in every other vertebrate. The genome of the icefish is not simply missing hemoglobin genes. It has been cleaned, gradually, systematically, of the entire support structure that hemoglobin required. Each lost protein relieved the organism of the cost of synthesizing it, the enzymatic machinery to assemble it, and the regulatory systems to manage it. The subtraction is cumulative and coherent. At 2 in the morning, the icefish has been motionless for hours. This is not inactivity, it is ambush. The icefish is a predator, feeding primarily on krill and small fish. but it hunts by waiting rather than pursuing. In complete darkness, its translucent body offers no silhouette to prey or predator. Any ambient bioluminescent light passing through the water penetrates directly through its flesh, casting no shadow on the sediment below. It achieves optical near invisibility simply by having no pigment to intercept photons. When prey passes within range, a short burst of the wide, fan-like pectoral fins closes the gap. The strike is brief. The return to rest is immediate. The icefish does not sustain effort. It applies it in the smallest possible dose and returns to its minimal energy state as quickly as possible. The broad pectoral fins serve a secondary function at rest. They distribute the fish's weight across a wide surface area of the sediment, minimizing the force per unit area against the soft mud. The fish does not sink or shift. It remains positioned exactly where it chose to wait, anchored by geometry rather than muscular effort, stable against the faint tidal microcurrents that move across the benthic plain. Around it, the glass sponges continue their silent filtration. The brittle stars make their imperceptible lateral adjustments. The deep water formation current delivers fresh oxygen from above. The temperature does not vary by a fraction of a degree. Nothing in this environment moves at a pace that would register as motion on human timescales. This is not a harsh environment. It is an extraordinarily stable one. The instability is at the surface. Storms, seasons, temperature swings, ice formation, and retreat. Down here, the water column absorbs all of it, and the benthic plain sits in a thermal and chemical equilibrium that has persisted season after season for millions of years. The icefish is motionless on the mud. Its white heart turns slowly. The clear plasma moves through wide channels with the quiet efficiency of a river with no gradient to push against. The antifreeze proteins hold their microscopic ice crystals in arrest. The mitochondria at the cell margins accept the oxygen that drifts across from the plasma and convert it steadily into the small, continuous expenditure of energy that keeping this body alive requires. Nothing is surplus. Nothing is struggling. The organism has arrived through 5 million years of iterative simplification at precisely the minimum configuration needed to persist in this environment indefinitely. The Southern Ocean delivers what it needs. The body uses what arrives. The rest is stillness. Whatever the day required of you, that's at the surface. Up there, with the ice and the wind and the polar dark, it doesn't come down here. Down here, the temperature is constant. The oxygen arrives continuously, and the heart turns at its own pace. Good night.